Welcome back. We're going to go over part two now of chapter six and talking about nutrition and the important things that the bacteria do. And we're going to talk about temperature related growth, ways that microorganisms function in the presence of these different types of oxygen conditions, and then other factors. We're going to talk about uh, important environmental pressures that they have to deal with, and then five types of associations that they have with their hosts, and some of them are good, and then my favorite topic on biofilms, and we'll get into that. So cardinal temperatures, that's the range of temperatures for growth of a given microbial species. So nothing to do with the North Carolina state bird. Cardinal temperature is just a range that a species can grow within. A minimum temperature is the lowest temperature that permits the growth. Maximum temperature is the highest temperature before proteins are denatured. Optimum temperature, which that's good to know, is an intermediate between the minimum and the maximum that promotes the best growth and metabolism. Psychrophiles, temperatures below. So sacro is cold. Um, Psychrophiles is cold liking, capable of growth at 0 degrees Celsius, obligate with respect to cold, and cannot grow above 20. So it has to be cold if it's an obligate. Storage, it's obligated, let's put it that way. Storage at refrigerator temperatures causes them to grow rather than inhibiting them. So if you ever wondered why you open the refrigerator door and things wave at you, well, it's probably a psychrophile. Natural habitats of psychrophile bacteria, fungi, and algae are lakes, rivers, snowfields, polar ice, and the deep ocean. None of which is a surprise. Uh, and they're rarely pathogenic, so I'm not going to worry about it. Psychrotrophs grow slowly in the cold, and that's why the refrigerators seem to do their duty, but have an optimum temperature between 15 and 30. Unfortunately, within that range, Staph aureus and Listeria monocytogenes are able to grow at refrigerator temperatures. So, I don't know if you remember reading about mother made her son eat leftover spaghetti, and it had been in there a while, I guess. And I think he ended up dying from Listeria monocytogenes. You have to have a lot of them to do it, but boy, that was upsetting to read that. So I think that should be a staple on all refrigerator doors, and kids unite for not eating leftovers. But hey, as a microbiologist, I know that Americans are so, well, the way they are, they throw away about half their food because of spoilage or whatever, and it'd be better just to make sure you just have on hand what you need. Mesophiles, majority of medically significant. They grow at the temperatures of 20 to 40. The average is around 30 something, which we're at 37 degrees, so there you go. And inhabit animals and plants as well as soil, water temperature, subtropical and tropical regions. Human pathogens have optimal temperatures between 30 and 40, which makes sense, degrees centigrade. Can survive short exposure to high temperatures but are normally mesophiles. So that thermoduric means something that can handle the heat, uh, without having the proteins denature. Common contaminants of heated or pasteurized foods. Examples of heat resistant endospore formers, of course, are Bacillus and Clostridium, which are two really nasty ones, of course. Thermophiles, like temperatures greater than 45, live in soil and water associated with volcanic activity, compost piles, and inhabitants directly exposed to the sun. Vary in heat requirements, but the growth range is around 45 to 80. Most eukaryotic forms can't survive above 60. Oddly enough, they identified a microorganism that was able to grow in thermal vents, And this particular organism, Thermophilus, was actually used, or has been used, and still is to this day, the particular DNA polymerase from that organism, because it is a thermophile, for PCR technology. So when we heat up the solution in order to amplify DNA, we use the DNA polymerase from an organism that's a thermophile. So pretty cool, pretty cool. So here is the group temperature ranges. You can see we've got various groups across the board there, and it's if you look at the high ones up here, the optimum was right at the very tippy top. You can see the optimum. Tippy top is a technical term, but don't write that one down. They're, you can see they're kind of sloped. Most of them are. It's I don't know exactly the nature of that, because when it gets to a point, I guess you're pushing the very edge of the proteins and then you start to fall apart. It would be my guess. But there's the names. There are the ranges that they grow at, and the optimum would be the peak up here, the forward peak. Pretty cool. That's a good graph to have. The atmospheric gases that influence microbial growth are oxygen and CO2. O2 has the greatest impact. You know, if you go in, I think I've already made reference to going into a retirement home and touching you have to ask for permission or you might get hit by an umbrella. The shins of some of the inmates, I mean folks that are living in those retirement homes, it'll be hot most likely, and the skin's gotten thicker. It's mainly because of oxygen stasis. Oxygen's not moving around as much because they're not moving around as much. And it's my public announcement always has been, if grandma's riding around in those little electric carts, get her out of it, unless she just absolutely cannot do it, because they need the exercise. It's important. Those things are important. O2 is an important respiratory gas and a powerful oxidizing agent. So when you see the heat going on in the legs there, it's usually due to the fact that it's toxic. And if it's not moving, so oxygen's not as toxic if we're moving and dealing with it, but if it's sitting around, then it's a problem. And so we want to deal with that. Microbes fall into one of three categories: those that use oxygen and detoxify it, those that can neither use oxygen nor detoxify it, and those who cannot use oxygen but can detoxify it. So the one in the middle here, those are the ones that I've dealt with. I can tell you really quick. There was a janitor, a really nice person, and he was cleaning up the lab that I was working in. It was a big manipulative hood. It was used for anaerobe growth. It's an anaerobic hood. And it was, instead of those real expensive ones are metal enclosed, this one was done with plastic. And he was mopping the floor, and the mop handle pierced the containment for that anaerobe chamber. And oh my gosh, you would, coming in on Monday, oh boy, that whole building smelled like an open toilet, a sewage treatment plant or something. It was just awful, and killed every strain I had growing. I was trying to prepare for a lab. As a graduate student, I did some teaching in the anaerobe lab. No one else would do it, and it paid pretty well, so I did it. And wow, stinky, but when it says it can neither use oxygen nor detoxify, it will kill them, and I can attest to that. Clostridium species gone. Oxygen enters cellular reactions and is transformed into toxic products. The singlet oxygen is an extremely reactive molecule. The electron transport chain, when we're breaking things down, when we crack water, that is actually a problem. And I call it like handling the hot potato, because it is a really hot potato. So you have to have means to deal with that, to convert that singlet oxygen. Another highly reactive is the superoxide ion, which is an O2 ion. Now, remember now, the word ion means that it's that unusual case where it it either gains or loses an electron, and then so it looks like we've gained one here because there's a negative symbol. Hydrogen peroxide, H2O2. There you go. Uh, it's toxic to cells and is used as a disinfectant. It's always stored in a brown bottle. Why do you think that is? Why do you think they store the hydrogen peroxide on the shelf in a brown bottle? You never see it in any other bottle. I heard you. Yep. You have to yell louder. It's kind of hard to hear you from wherever you're at. I'm in Apex. Uh, Hydrogen peroxide breaks down in the presence of oxygen, light, so they have to use a brown bottle to kind of hamper that. But one of the tricks I do is I want to get the freshest hydrogen peroxide. A lot of times I'm using it for an experiment, but I just go and buy a little bit at a time. And so I would go to the store and grab the bottle in the very back because those are the newest products in the back. They want to get rid of the old ones in the front. I think you already know that. But the idea is, over time, if you let hydrogen peroxide sit on the shelf, even in a brown bottle, it'll eventually turn to water. And I just don't want to be spending a lot of money for just a little thing of water and not get the benefit of the oxidation there. It's toxic to cells. It's a great disinfectant. It's a really good one to use around ears in children. If you have ear infection in children, it's pretty innocuous. You can, of course, get a doctor's okay, but it's a good cleaning agent. I use it a lot of times if we had animals that came in with that had huge fights. I mean, they're all torn up and their skin's hanging out and all that stuff. And we would lavage it or just clean out the insides with all the dirt and grass and God knows what else was in there with hydrogen peroxide, and it worked really well. Hydrogen radical, which when we break down water into hydrogen and OH, it's highly reactive. The OH part is, and it's looking for a way to balance its charge there to compensate all of the octet rule. Most cells have developed enzymes that scavenge and neutralize reactive oxygen byproducts because the ones that don't survive very long. So it's a pretty good motivator. So we have two-step process requires two enzymes. One of ones that when I was at NC State as an undergraduate, there was a lot of research going on with superoxide dismutase, and that enzyme drives the oxygen, these radicals which are highly reactive, and hydrogen, and produces hydrogen peroxide and oxygen. And then catalase takes the hydrogen peroxide and brings it to water and oxygen. So catalase causes bubbles to form. If you break down hydrogen peroxide, it just starts to bubble. Catalase will do that. We actually use that as a test in the lab to see if a catalase is present or not, and a positive test is it bubbles and it does it immediately. So it's a great test for that to see if catalase is present. Superoxide ion is converted into hydrogen peroxide by superoxide dismutase, and then that peroxide is broken down by catalase. So organisms can do that. Aerobes can use gaseous oxygen in their metabolism and possess the enzymes needed to process toxic oxygen products. An organism that cannot grow without oxygen is an obligate aerobe. Makes sense. It can't grow without it, so we're sort of like an obligate aerobe. You block our breathing, and well, not good. Most fungi, protozoa, and many bacteria such as bacillus and mycobacterium species are like that. Microaerophiles are harmed by normal atmospheric concentrations of oxygen but require a small amount of it for its metabolism. So in those environments, it's greatly reduced from atmospheric oxygen. So that Helicobacter pylori that causes ulcers, it sits a little bit higher up than the sphincter for the top part of the stomach. And Borrelia burgdorferi, that's one of my hated organisms. It knows it too. Borrelia likes to hang out in poorly vascularized regions, obviously where blood's not pumping, and of course we get a little bit of the diffusion coming out from the capillaries and things. So Borrelia loves to hang out in joints and things like that because, well, It's a microaerophile, and it prefers those sorts of little nasty niches, and that's why one of the reasons why it's so hard to get rid of Lyme disease agent, because of where it likes to hang out. Of course, when you take an antibiotic, if it's poorly vascularized, means that once your antibiotic gets into your blood and things, it's going to take a long time to get sufficient amount of that to those poorly vascularized regions, and Borrelia knows it. It read the transcript. No, it by just through just normal processes, of course. If it's not there, it's going to survive, and there you go. Faculty, the faculty that no, the facultative anaerobes do not require oxygen for metabolism, but use it when it's present. So uh, many gram-negative bacteria and staph are facultative anaerobes. E. coli is a good example. Staph, is something like that. Anaerobes lack the metabolic enzyme systems for using oxygen, so obligate anaerobes also lack enzymes for producing toxic oxygen and die in its presence. So they've evolved without having those enzymes to break it down for oxygen. So, but not good. So, uh, intestinal, many oral bacteria. Air tolerant anaerobes don't utilize oxygen but can survive. And we have, they're not harmed by oxygen mainly because they possess alternative mechanisms for breaking down peroxides and superoxides. But lactobacilli, which is a probiotic type bacteria, streptococcus, and clostridial species are aerotolerant. Okay, capnophiles. Now, capnal just means carbon dioxide. It's well, technically, it's not what it says in Latin, but that's what it means. A bluish color. It kind of brings back all these. Yeah, Latin. It's 3 years of Latin. Boy. Anyhow, organisms that grow best at higher CO2 tension than normal and present in atmosphere. It's important in the initial isolation of the following organisms: Neisseria, and the one that there's Neisseria meningitidis is a real problem. We see this in teenagers that are sharing, like colleges and the like. Probably won't see a higher incidence of rate of that during the COVID times here because you were not sharing bathrooms with other folks, and that should go down a little bit. It usually presents as a severe headache that won't go away, and physicians know to look for that age group and college students because it can be very serious. It can be life threatening pretty quickly, and if it's misdiagnosed, that's not good. So, anyhow, there's a reason I mention it. Brucella, undulant fever, brucellosis is a way to look at that. Streptococcus pneumoniae causes a pneumonia. pH, defined as the degree of acidity or alkalinity of a solution, expressed by the pH scale. Now, pH, I always like to tell my students, pH, think of it as percent hydrogen, because that's really what it is. At 100% hydrogen, we've got an acid state, 0. At 0% hydrogen, we've got the high pH of 14. And so the balance in between the two is pure water. And so that's a pH of 7. So the hydrogen percent, you can imagine, I'll give you a few seconds, 50%. So that's halfway. And so what is the other 50? Well, when you break water down, you get hydrogen. What's the other part? Hydroxide, the OH group. So you have 100% of OH concentration at 14, and you have 0 at 0 pH of hydroxide. So that's why I like to look at it as percent hydrogen. At 100%, it's 0 pH. At 0% of hydrogen, it's 14. So they're inversely proportional. And both are at 50%, then we're dealing with a pH of 7. And then you can work from that. But the majority of organisms live and grow at habitats between pH 6 and 8. So what is the pH of our blood, the human blood? Give you a few seconds. You have to yell it really loud. 7.4? Yeah. And if that pH changes to 7.2, not compatible with life. 7.8, not compatible with life. What do you think really drives that pH change and why it's important to maintain the 7.4? That's a really important question. And I can give you a hint that proteins change their shape as you change pH, because certain things protonize and all that, and it will change its shape and to the point where it won't function. And if a certain protein that causes, like, say, the breathing center or the heart to beat, and that pH change causes it to change its shape, and then the heart doesn't beat, or we don't breathe, or that sort of thing. So it affects various proteins that may be absolutely incredibly important enzymes or whatever. Acidophiles, organisms that thrive in acidic environments. Euglena mutabilis. Mutabilis. There you go. Finally, spit it out there, Dr. V. Grows in acid pools between 0 and 1. I've never worked with that organism. Thermoplasma lives in coal piles, 1 to 2. Psychrophiles lives at a pH of zero point 0.7. Wow. Can grow at pH 0. Jeez Louise, amazing. Many molds, I would believe it. Molds and yeast tolerate acid, more common spoilage. Pickled foods, yeah. I think molds grow on anything. Alkalinophiles, those like to go at the other end of the spectrum. Natromonas live in hot pools and pH is 12. Proteus can create an alkaline condition, neutralize urine, and colonize and infect the urinary system. Proteus mirabilis and other organisms like that. Osmophiles live in habitats with high solute concentration. These are osmotic pressures. So osmophiles like high solute concentration, so they can handle pretty high osmotic. Halophiles prefer a higher concentration of salt. An obligate halophile, like Halobacterium or Halococcus, grow normally at solution 25% sodium chloride. Wow. Facultative halophiles are remarkably resistant to salt, although they don't normally reside in high salt. Staph aureus can grow in salt media from 0.1% to 20. So that we actually take advantage of that and use a type of media that has a lot of salt in it. And we'll talk about that when we get to the lab. Radiation. Phototrophs use visible light rays as an energy source. Non-photosynthetic microbes tend to be damaged by the toxic oxygen products produced by contact with light. Some microbe species produce a yellow carotenoid pigment to absorb and dismantle the oxygen, sort of like our melanin, and it causes skin to tan and that sort of thing, and or get darker depending on the skin tone. UV and ionizing radiation can be used to microbial control. One of the things that's not mentioned here is it damages DNA, and we get, as a result of UV radiation, thymine dimers, and then that'll cause frame shift mutations, and that's usually lethal. Biofilms exist under pressure that range from a few times to over 1,000 times the pressure of the atmosphere, and so every 3 feet of water is 1 atmosphere. So that's 3 feet. Is that right? 30 feet. Wow, it's been a while since I've done my dive tables. 30 feet. Yeah, every 30 feet is an atmosphere. And so you can go down to about 100 feet. That's 3 atmospheres. That's I believe that's right. Anyhow, the idea is it's pressure because of the weight of the water. The bacteria are so strictly adapted to high pressures that they'll rupture when exposed to normal. And so, as it is, they have to adapt systems to manage those sorts of things. You change it too quickly and it kills them. Other organisms, rarest microbes, shared habitats, associations between similar and dissimilar, associations with multicellular such as animals or plants, which is really interesting to me, is the associations between things, the interactions. They can be beneficial, harmful, or have no particular effect. I mean, just walking outside And you have no clue when you're stepping on the grass how much of all of these associations are going on that for water absorption. And it's just amazing, all these associations that go on, and we just have no clue or just take it for granted. A symbiosis, general term denotes a situation in which two organisms live together in close relationship, or symbionts. Uh, Three different types of symbiosis is mutualism. Organisms live in an obligatory but mutually beneficial, so it's a win-win. Commensalism is a partner called the commensal receives benefits while its partner is neither harmed nor benefited, so it's sort of like a win and who cares. And then parasitism is a relationship in which the host organism provides the parasitic microbe with nutrients and habitat, and the host suffers from the relationship. So that's a win-lose. And so those are the three. Antagonism is an association between free living species that arise when members of a community compete. Antibiosis, production of inhibitory compounds such as antibiotics in the surrounding. Now, antibiosis is something that probiotics use to try to control or police our gut. And that's why we want them there, is to try to make sure that they're the ones present and they'll destroy other microbes that might be of harm. So that's why I'm a big believer of probiotics. The first microbe has a competitive advantage by increasing the space and nutrients available to it. Common in the soil, where mixed communities compete for space, food, water, and other nutrients, and it's amazing. It's pros and cons. Synergism, interrelationship between two organisms that benefit them but not necessarily for survival. Together, the participants cooperate to produce a result that neither of them could do alone. So they're doing something beyond the capability of either. And that's what a biofilm does. Gum disease, dental caries, clousals, whatever. We got some dentists in the audience, I think. And some bloodstream infections involved in mixed infections are examples of bacteria that interact synergistically. And so they think that perhaps other diseases could be formed as a result of the poor gum health. Alzheimer's, they're thinking that they're seeing a direct correlation of gum disease and dementia, and really strange, but there you go. Biofilms, the epitome of synergy, my favorite topic of all times. I have a paper I would like to give you, and I'll post that in the section on biofilms. I think I already gave that out for reading, if I'm not mistaken. I'll have to go back and check. I'm sitting in my man cave here. Oh, no, I'm not supposed to say that. My temporary office to ride through the storm here. If pioneer colonizer attaches, then microbes come, and because it's kind of breaking the ground, others start to colonize, then start sharing. And then once they start sharing, other things start to grow, and before you know it, you've got a whole community with phones and toilets and porta potties and God knows what else, and they're all working together. So anyhow, that's the important part. So when certain organisms come close together, they trigger other things and are activated, and free living bacteria and the like, the genes get turned on or turned off, and there's a whole series of research that's going on about that right now with biofilms. Very interesting research, and there's some schools that are working on that. So that's what I wanted to complete with the part 2 here, and we successfully did that. Next time we're going to be looking at how doubling time and how cultures are grown, and I think it's an important section, so I didn't want to rush through it. We'll handle that as part 3, and then we'll be done. I believe this is where we're at, and I'll finish that up, and you should have all of these shortly.